So as we start our aquaponics design projects, one of the things that you've already identified, I think, is the need to understand photosynthesis and how we might be able to optimize different components or different variables or factors of the photosynthetic rates, which really means that you need a wealth of background knowledge about what's going on in photosynthesis. And so I'm going to make a little video here to give you an overview, and in all likelihood, it's probably going to be in two parts. So the first part is going to kind of help explain some of the content that's in section 2.9. What was it, 2.9 of your book, which starts on page 129 and on page 389. And the part that's on page 389 is likely going to be in the second video. And so really what you need to be able to do is define what this term means, what photosynthesis means. And really, it is an enzymatically controlled process in which carbon compounds are made using light energy. Okay, so let me write that out for you. So this is an enzymatically controlled process in which carbon compounds are made with light energy. All right, so that's all good and well, and I hope that, maybe you ask some more questions here, like what the heck does enzymatically mean? I hope that you add that to things that you might need to know if you don't know what that means, or what carbon compounds are, or really what light energy is, because part of the thing is that this definition makes it sound like light energy is all that you need to make carbon compounds, which is absolutely false, right? So I hope that you remember, and I'm going to jot down this equation, the general basic photosynthetic reaction. And just like with the puzzle pieces that we did in class, it's going to get more complex. The longer I talk about it, the more complex it's going to be for you. So I hope you remember that, plants need CO2 and water and light. And I'm going to put that over the arrow because it's matter and not energy. And what the plants make in this photosynthetic process is glucose, C6H12O, that's an O6, plus oxygen gas, or O2, right? So that's the general equation. It's not balanced in any way, so let me go ahead and balance it. I got 6 carbons here, so I need 6 carbons in carbon dioxide. I've got 12 hydrogens, but I've only got 2 on the left side, so I put a 6 there. I've got my 12 hydrogens, and hey, look at that. It's almost balanced, but I've got 6 oxygens over there, 12 oxygens over here. I've got 6 oxygens over there, but if I add a little 6 here, now I've got a nice balanced net reaction. But what I want you to understand and not be misled by is that that's not the only thing going on here, right? So these are the reactants, and these are the products of this reaction overall, but there are tons and tons of intermediate byproducts within the process, because it's not a simple process, which is so cool, right? So this is a really complicated process. It's the basis of most of our food webs, definitely ones that we're dependent on, and it's really, really complicated, and we continue to learn more about this process. So a couple of things that you should, I hope, remember is that this process is happening in chloroplasts. Often, although chloroplasts is not the only place it happens, right? Chloroplasts are a specialized organelle within some plant cells. You might make the assumption that all plant cells have chloroplasts. They do not. Only the leaves and sometimes stems, which are green, have the chloroplasts, which have that green pigment chlorophyll, right? So roots don't have that. The trunk of a tree doesn't have chloroplasts. Instead, they have other specialized organelles. We'll learn more about those organelles, but they store the byproducts, or really all this glucose that's building up in the plant. So, or other forms of that glucose that get built up. So, what we're going to do is kind of draw as best as I can here. I'm going to pick my, I'm going to pick yellow, and what we're going to do is draw kind of an oval here. And what I'm going to do is tell you that that's a chloroplast. And actually what I'm going to do is also tell you that it has a double phospholipid bilayer. Okay? A double phospholipid bilayer. And so this, I'm going to do two arrows there. So I'm going to label this as a double phospholipid bilayer. Now, it has two phospholipid bilayers. I hope you remember phospholipid bilayers from 9th grade. But it's two of them because of something called the endosymbiotic theory. Or at least that's our explanation of it. We don't know that's absolutely true, but it is a theory, a scientific theory based on lots of data, which we'll talk about later. It's due to the endosymbiotic theory. Okay? So within this chloroplast, I guess I should label this as a chloroplast, huh? So this thing is a chloroplast, a single chloroplast within a single cell, within a single leaf, etc. So here's my chloroplast. Inside that chloroplast, we have these structures called thylakoids, and I'll spell that in a second. So they're essentially these stacks, right? They kind of look like stacks of pancakes. And there's several of them. They're all throughout. And they have tons and tons of membrane, right? So they have lots of edges. So they have a high surface area to volume ratio. But these thylakoids, I'll spell it, thylakoid. These thylakoids, in their membranes, house, so in membranes, there's chlorophyll. All right? So that chlorophyll is really what's absorbing a lot of energy. So this chlorophyll, let's see if I can scroll down here a little bit. All right, so the chlorophyll absorbs some of this energy. So if we have, I'm going to say light, it doesn't have to be sun, right? So that light is going to excite electrons in the thylakoid. Those electrons are going to be used in another process. But one of the things that we mentioned earlier is water. Water is really important for these leaves. But remember, and you see kids do this all the time, especially little kids. They try to water plants by pouring water on the leaves. But we know, I hope you know, that the plants absorb the water out of the soil. That moves up the plant through capillary action and gets to the chloroplasts in the leaves. All through the roots, right? So water is coming up from the soil, and that water, the H2O, will get split into a couple things. And again, this is an oversimplification. The next video is going to go into a little bit more detail about this. That water is going to get split into a couple of things. So light is going to go through this reaction called photolysis. Photolysis. Photo means light. Lysis means to break. Jot that down. Lysis is to break. So what happens is that the light breaks the water, and out comes oxygen. All right? Out comes oxygen gas. So again, this isn't balanced. We've actually got 6 waters here, right? So we get some oxygen that's coming out, and we're just following the atoms here. The hydrogens actually are going to be quite important because the hydrogens are getting added to this molecule called NADPH. And there's the H, the hydrogens. NADPH that came from the water. The energy that held the covalent bonds of water have been broken by this light energy, and it has been captured, we'll talk about this in a little bit more detail in the next video, as ATP. ATP, you should jot down, ATP is adenosine triphosphate, our most common energy molecule. And then what that reaction does is it goes through, those things go through a series of another reaction. And again, we're going to go into more detail about this. It's called the Calvin cycle. Okay? The Calvin cycle. So now we've got oxygen taken care of. That I'm not going to put a check mark there, but you could put a check mark. We talked about oxygen being a product, talked about light being a reactant, and water being a reactant. But we haven't talked about CO2 and glucose yet. So this is really where CO2 comes into play. So CO2 enters the Calvin cycle. The carbon, the oxygen, and the hydrogen that originally came from the water leaves the Calvin cycle in the form of C6H12O6, or glucose. And the molecule, and it uses some of the energy in the ATP. So what ends up happening is that leaves, and so we end up with NADP plus and a adenosine diphosphate plus a lone phosphate. So the bond between the two phosphate groups to the third phosphate group has been broken. And that energy was released and used to combine this larger molecule, and now we've got all the reactants we need. Now what happens with that glucose is another whole story, right? So that glucose can go through a series of reactions where a lot of glucose get bonded together to make other carbohydrates like cellulose or starch or sucrose, right? Or other disaccharides. And we'll talk a little bit more about that as well. But this connects very well with those other larger units, right? So this is the overall process, but there are two major reactions here. And we're going to label them so you have some context for them. So the first one, let me pick a better color here. So the first one is over here. And remember, I'm one of the limitations of my drawing is that I put thylakoids over here on the left, but really these stacks of thylakoids, or granum, the stacks are called granum, Granum, or multiple granums is really grana. If you take Latin, you know that it's Grana for plural granum. But that first reaction, and put a 1 there, is called the light dependent reaction. Okay? So what that really tells you is that it needs light, and it happens so requires light, happens in the membrane of the thylakoid, and this is really where photolysis happens. Okay? Oops. Now later we will talk about two things, and I'll jot them down here so you have them. There are two reactions happening. There's or two kinds of things. So there's photosystem 2, which is abbreviated this way, and then photosystem 1. And I'm gonna leave that as a preview for the next video so you understand really what the context is of that. Now the Calvin cycle, so the Calvin cycle, I'll write that again, Calvin cycle, it goes by a couple other names, right? So sometimes it's called the light independent reaction, which is a little misleading. Because it does indicate that it doesn't use light directly, but it does need NADPH and ATP, which are byproducts of the light dependent reaction. So if a chloroplast were to go for long periods of time without light, then the NADPH and ATP would be used up, and therefore the light-independent reaction wouldn't be able to move forward because it needs things from the light-dependent reaction. So photosynthesis can actually occur, or at least parts of it can occur at night when there is no light, as long as the reaction in the light-dependent reaction is happening at such a high rate that it builds up the amount of NADPH and ATP so that those can continuously be used overnight in the Calvin cycle. But there's another word that is used called the dark cycle to describe this thing. And that is really a misleading term because being light independent and dark are not the same thing. Being light independent means that whether there's light or not, this reaction can move forward. Dark cycle implies, as opposed to the light dependent cycle, that it only happens at night. And that's not true. That's not what light independent means. So it can get a little bit confusing. But what you should know is that this is sometimes referred to as carbon fixation. So the carbon in the CO2, an inorganic compound. CO2 is an inorganic. Organic really means carbon containing, but it can be a carbon oxide kind of thing. So CO2, because it's missing one of the other key elements, hydrogen, it's not considered organic. But what photosynthesis does is takes this inorganic carbon molecule, CO2, and turns it into glucose, which is definitely organic. Okay? And is a building block of living things. So carbon fixation is what's happening here, and the location of it is the stroma. Okay? Stroma. We're going to talk about another vocab word related to plants and leaves later called stomata. And it does relate to photosynthesis, but we're not going to talk about that quite yet. All right, so that being said, that is the process, the overview of photosynthesis. I see that I'm at 17 minutes. I apologize. I'm going to keep going here. We're going to talk a little bit about the light itself, right? So I'm going to focus in on light. And let me make a new page here. So when we talk about light, what we're really talking about for photosynthesis is visible light. So not all light is visible. I hope you know that. So we talk about the visible light. We're talking about, I hope you know this, Roy G. Biv, right? Red, orange, yellow, green, blue, indigo, violet. The colors of the rainbow. These are the visible light spectrums that we have. And they have a range. What makes them different colors is the wavelength, right? So they have a range of wavelengths. And these wavelengths are between 400 and 700 nanometers. Okay? So if you don't know how many nanometers there are in a meter, I want you to look that up. You need to look that up so you understand the context and the real value of this information, right? So, and we're going to point out that red has the longest wavelength, okay? Longest wavelength. So that's about 700. And violet has the shortest wavelength, around 400. Okay, if you and the thing here is that you need to make sure you understand that these wavelengths are really key because there are pigments like chlorophyll. That's one of at least 4 different pigments in leaves that absorb light and then are able to transfer some of the energy from the photosystems 2 and then 1 into these organic molecules that are made through photosynthesis. So what we're going to talk about very quickly is some... actually, let me see if I should do this. Yeah, let's go ahead and talk about absorption and action spectra. And actually, I'm not gonna talk too much about this. I'm gonna ask you to read a little bit in your book. Absorption and action spectra. So I want you to look at page 133. And there's a little blurb with a purple background. I want you to make sure you read the text and you look at the graphs and you understand what's going on. But what you will see is that there's a dip. The rate of photosynthesis is lowest with green light, right? So with green light, we have the lowest rate of photosynthesis and the percentage of light being absorbed in the plants. And I want you to think about that. I want you to share your answers about that, and so that we can see what you're thinking, right? I think that once you understand it, it will make perfect sense to you, and you don't need to. We won't have to worry about that again. So the other thing that you should think a little bit about and look over is the limiting factors, which starts on page 136. So it's about temperature, light intensity, and carbon dioxide concentration, and how those things affect the rate of photosynthesis. And I'm actually going to do a quick graph of this. So I'm just going to kind of do three graphs without any tick marks. So it's just a sketch of the graph, and we're going to try to offer some explanation. So not only describe the graph, but offer explanations for why the graphs look the way they do. So if we're looking at, I'm just going to abbreviate this as rate of photosynthesis and temperature, what you should predict is that because temperature is a measurement of molecules of, well, if it's air temperature, the molecules of the air that are bumping into each other. And if we're looking at this diagram from before, we know that CO2 is a molecule in the air, and it will make up a large part of the glucose that's being made. But the faster you can move the carbon dioxide and get it into the leaves of the plant through the stomata, the holes in the bottom of the leaves, to the chloroplasts, the more or the faster the rate of photosynthesis you can get. So as temperature increases, the rate of photosynthesis increases. It hits some optimal level, and then it actually comes crashing down. Okay, and the first thing that we should make sure that we understand is that as temperature increases, the reason that the rate goes up is that molecules move faster, and I'm running out of space, but I want you to add, as the molecules move faster, CO2 and oxygen can diffuse in and out of the leaves, right? So there's some optimal level here, and the reason it's coming crashing down is twofold. There are two reasons for this. If you look back at the definition for photosynthesis, it's enzymatically controlled. So beyond some optimal temperature, enzymes are denatured. All right? So enzymes, if you don't know, are proteins. They're specialized proteins that speed up reactions. They allow them to happen efficiently without using lots of energy. But when you heat them up, the proteins change shape. And you know this, like for when you cook an egg. The white of an egg is not actually white until you cook it. And the shape of the protein, that albumin that's in the white of the egg, is changing its shape and reflecting light differently after you've changed the shape of it. Well, when enzymes are denatured, they can't be undenatured. They don't change shapes back. And so when the enzymes are no longer working, the rate of photosynthesis decreases. So that's one reason. Another reason is that the stomata close. So to prevent water loss. All right, so prevent water loss. So the stomata, again, are holes underneath the leaves. What they allow for is CO2 to come into the leaves and oxygen to leave the leaves. But when it's too hot, water will also leave through the stomata. And that's not so good for plants, right? So plants can't go find another source of water. That's why they have to store a lot of water in their large central vacuoles within their cells. But if they're losing so much water, they're actually going to close those pores, shut down photosynthesis, so that they can save the water within their cells. And so that will also cause the photosynthetic rate to come crashing down. Now, the key here, though, is that the optimal temperature, whatever that is, may differ based on the environment and the species of plant and the adaptations of the plant. So, for example, some plants have a really waxy coating, like think of aloe vera that lives in a desert, so that it does not lose a lot of water. Or a cactus with its needles have really, those needles are its leaves in case you didn't know, and those leaves have very little surface area, so it loses less water in that arid temperature. Okay? So this can move. That's why I didn't put any scale down here. So I hope you can see how temperature is a limiting factor here. We also need to make sure you understand light intensity. So the effect of light intensity. I'm just going to put light intensity on the x-axis. I'm not going to rewrite rate of photosynthesis on the y-axis, but you can go ahead and do that. I've run out of space. So as light intensity increases, you're providing more and more energy to the system, so the rate of photosynthesis can increase, but then it plateaus, right? So it reaches some maximal level and then plateaus. And the reason that it plateaus is because if you're only increasing the light intensity, there's not enough of the other reactants. So if we're not adding any more CO2 or water, it's not going to drive that reaction upward forever, right? I think that makes sense. I hope that makes sense to you. And the other one is carbon dioxide concentration, right? So carbon dioxide concentration, because it's a reactant, if you add more of it, it will increase the rate of photosynthesis up to some level and then plateau for the same reason light intensity plateaued. There's not enough of the other reactants, right? So if you're trying to optimize the rate of photosynthesis for your plants, depending on the species, you might want to try to find the sweet spot of temperature, the optimal temperature. You might want to find the optimal intensity, but it plateaus, so really your optimal intensity could be anywhere in here. But if we're looking at what the word optimize means, if you can burn less energy and waste the least amount of light intensity and yet have the maximal yield of photosynthesis, which would be right about here, you can be a lot more efficient, right? And same with, I didn't put this, CO2 concentration. If I pick this spot right here, then I'm not wasting the amount of CO2, resources, and other environmental factors. So it's important to know what the data looks like so that you can provide your plants the optimal. And so really it's going to be really important that you pick your species of plants or vegetables very quickly. Okay? And I'm going to stop there because I see I'm at 30 minutes.